All right. Well, let me make sure I have everybody. I have another person in the room who's entered, and I think that might include near all of our participants. So let's get started. Welcome everyone again. My name is Joan Shank, and I'm the Chief Education Program Officer at the Wake Forest Institute for Regenerative Medicine. And this afternoon, we're really excited to connect with all of you and provide an informational session about the Summer Scholars Program, from just the basics, how to apply to the program, elements of the program, and any questions you might have about that process. That's just kind of, the mechanics. But what I'm also very excited to do is introduce you to two of our faculty mentors who participate in the program. We have a total of 35 faculty mentors for the Summer Scholars Program. This year, we'll be recruiting for about 20 slots. So we have more projects than we have students. So gives us a lot of flexibility in really matching what you're interested in the field. But you'll also be introduced to one of our former scholars, Emma Warner, who is a biomedical engineering student who joined us last summer from University of Miami, and she's done some pretty exciting things that I'm not going to steal her thunder that she'll share with you. And then we also have Dr. Josh Maxwell, who is an assistant professor with us at the Institute for Reg Med. He has a PhD in cell and molecular physiology at Loyola University. I'm not going to give a lot of background information. You can see their bios here, but they're going to talk about their background and their research during their presentations. But Josh does a lot of very interesting, cutting edge things focused on using stem cell therapies and other approaches for heart failure, predominantly in pediatrics, but in other areas as well. And Professor Graça Almeida, she's an MD PhD, and she has, her work has looked at cell and delivery platforms, predominantly for genetic and immune diseases. So she has some really very exciting work that she'll share with you. But this is really geared again toward their participation, their research, of course, but their participation in the program as mentors. And then following Dr. Almeida Parada's presentation, you'll hear from Emma Warner, who I referred to earlier. And again, Emma, thank you for joining us this afternoon. She actually was a summer scholar who worked with Dr. Almeida Parada this summer. So with all of that said, let's get cranking here. And I am going to kind of kick off the session. And following at the end of our presentations, what's most important is that we provide each of you an opportunity to interact with all of us and ask questions about the program, about the application process, anything that you might like. But here we are, as I mentioned, this is about the Summer Scholars Program. We're accepting applications now. I'm assuming that many of you are focused on developing your application. That is an online application process, and the deadline is January 5th. The program dates for this coming summer is May 1st to August 4th. We are located in Winston-Salem, North Carolina.
John, sorry, you're fading in and out. I don't know if it's just me or everyone is having trouble. When you look to the side, we stop hearing you.
Okay. You can hear my dogs really nicely though when okay, well, I'm going to climb into my laptop a little bit more here. So is this better? Excellent, excellent. Okay, so what I first want to do is just introduce you a little bit to our institute. We are a very large institute of regenerative medicine. We're not a virtual institute. Everything is under one roof. So all of our faculty are under one roof. We do team research. We cover all of the areas from enabling technologies, stem cell approaches, biomaterials, on and on and on, but it is all under one roof and you'll actually be interacting with the whole team. The mission of our research institute is to improve patients' lives through regenerative medicine. We are a globally focused and engaged institute with collaborations all across the United States and the globe. So when you are here at the institute, you're going to meet with our faculty, but we have people here all the time working. Our mission is supported by a number of key objectives, and the area that we're going to focus on today is in order for us to be able to, well into the future, to get these therapies and treatments and continue to do new basic research, is that we have to train and educate the next generation of folks who are going to continue to participate in the field, bring these things to patients, and develop new areas of research. We have a really strong portfolio of education that is sectored into 3 cores, graduate and postgraduate, undergraduate and workforce development, and K through 12 in the community. Today, we're focused on our undergraduate core and specifically our Summer Scholars Program. You know, as undergraduate students participating in this session, you know, I think what you're really interested in is moving beyond the classroom and really working in a real world, real life environment, conducting research. And we believe that that really is one of the richest ways that you can become involved and extend your classroom learning. So we've been doing this at the Institute for a very long time. Dr. Anthony Atala is the director of the Institute. I'm assuming that many of you, all of you on the call, are very aware of him. He's had TED Talks that have been viewed around the globe, and he's truly a pioneer in the field, and we're all excited to be a member of his team. He's actually been providing the undergraduate summer scholar program since inception. So we've been doing this since day one. And we've had more than 700 undergraduate students who have joined us during the summer and academic year, 283 that have been competitively selected. I can share these slides later, but we're really very proud of the program and the impact it's had on students, helping them figure out what they want to do next. Many students have actually near 80% of our students that come with us go into PhD or MD programs, and they come to us from across the US, across the nation, etc. So these are some photos, not all of all 24, but of many of our 2022 summer scholars who joined us last year. And we're really looking forward to developing, you know, to our 2023 cohort. Again, the program dates are May 31st to Friday, August 4th. You'll engage in team-based research, so you'll have a primary faculty mentor. You also have a supervisor. Our team works across with many other investigators, so you may be in projects that are multi-PI led as well. You do receive a stipend of $5,500 for the 10-week period, and you also participate in a number of short courses, seminars, workshops on different topics in the field, so you're exposed not just to your area, but to other areas. You also participate in a final research day where you present your work in an oral format and in a poster session. Many of our students, and you'll hear from Emma later, are also coached and encouraged to present at other scientific meetings. Near 60% of our participating scholars do that. That's really very exciting. We're a pretty fun group, we think, and maybe Emma can talk about that as well. You know, our students develop lifelong friendships and as colleagues and as friends after joining us in the summer. And we find that really exciting as well. We also do provide graduate and medical school advising, so you can learn about opportunities here as well at the medical school and in our PhD programs. So here's our timeline. The deadline is January 15th. It's an online application. We'll start notifying near the end of February if you've been successful with your application. Everyone will be notified by toward the end of March. Some questions, and we can talk more about this at the end. But all of this information is on our website, very detailed FAQs about the application. You do, there is an essay portion, and you know, just be real. And I'm looking forward to your questions about that specifically, and maybe Emma can even chat about the essays, the application, and how she approached it. We do need letters of recommendation and transcripts. So 2 letters of recommendation and your transcripts are to be sent to us. Here's the minimum GPA. Do you have to be a US citizen to apply? While most of our students are US citizens, we do have international students who join us as well. The only requirement is that you are attending a US university and you have your J1 or F1 visa already. Age restrictions, you do have to be 18 years of age. And at application time, you have to have completed 2 semesters of undergraduate education by start of the program. Do you need prior experience? Absolutely not. While some of our students do have experience, some exposure to this field as an undergraduate student, it's not required. It's an undergraduate program, and our goal is to expose you. But you certainly should be able to express your excitement, your interest in your application. I think that would be pretty important. We do have housing, and maybe Emma can even talk about that as well, that we provide pretty swanky apartments on the Renolda campus. So it's dormitory style. So housing is available. There is a shuttle. There is a little bit of travel involved, but nothing far. Travel expenses are not provided. And again, you will be receive a $5,500 stipend. So this is a list of where our summer scholars came from last year. You might recognize your institute here, but we've literally had students from so many institutes across the US join us each summer. And a list of our faculty on our website. You can find all of our faculty and kind of dive into their research interests. At WFIRM, it's a multidisciplinary field. It takes a team. And so, you know, we invite you to be part of our team. And with that said, I am really excited to introduce all of you to our faculty mentor, Dr. Josh Maxwell, who's going to introduce you to his work, as well as some information on the student that he mentored this year. So Dr. Maxwell, if you can get rolling, I am going to give the slide control over to you. You can just advance that. I've done that, yay. So you can get rocking and rolling, and I'll look forward to listening to your presentation. Thank you.
Thank you, Joan. It's great to be here with everybody. I'm going to give you a little introduction to what our research focus is in our lab, and then talk a little bit about the program and some of the goals that we have for that for all of you that apply and come here. So the focus of my lab is on looking at cardiac regeneration and repair, usually in the face of heart failure or after myocardial infarction or heart attack. We actually focus on pediatric dysfunction. So a lot of kids with congenital heart defects actually end up getting right ventricular heart failure fairly early in their life, need cardiac transplant. That's a pretty tough procedure, and tissues aren't always available. So what we want to do is use these different cell-based therapies to actually kind of restore cardiac function and prolong the time that these kids can go without needing a cardiac transplant. So building off of that, some of the things that we do are use animal models to study the molecular basis of right ventricular heart failure, and also use these animal models as well to test our stem cell therapies. So we kind of have three different pillars of research in our lab. The first is going to be looking at the mechanisms of exogenous stem or progenitor cell effect on heart function and heart repair. So that's going to be a lot of our cell-based therapies that we use to restore cardiac function. The next, we do a lot of confocal calcium imaging and live cell cardiac myocyte imaging to look at exactly what goes wrong in these different disease states, such as heart failure or after myocardial infarction. So what we do there is we have different animal models of disease, then we actually isolate the primary myocytes from those hearts and study their function in the lab. And then the third aspect of our research is using a really cool cell model known as induced pluripotent stem cells that we derive into cardiac myocytes. And we've been using those as a drug screening platform right now, looking for new antiarrhythmic drugs. So to talk a little bit first about the stem cell mediated therapy, we really have a couple different cell types that we look at in the lab. And really there is no cardiac stem cell. So this opens us up to a lot of different possibilities on what cell type might actually be the best to repair or restore cardiac function after disease. Some of those different options that we have might be bone marrow derived cells, endothelial progenitor cells, mesenchymal stem cells, these cardiac stem or progenitor cells known as CPCs, and then of course the induced pluripotent stem cells as well. And a lot of the ideas behind these stem cell therapies is that these cells actually don't become cardiac myocytes, but we think that they act through some sort of other mechanism that's not a direct differentiation into a cell type that might help the heart. So what we think is actually that a lot of these cells are actually releasing factors such as different growth factors, exosomes, which contain microRNAs, that we think are actually acting on the native cardiac tissue to restore the function. And I'll talk a little bit about that more in the next slide. But I wanted to highlight here is that, a lot of these cell types have actually been entered into clinical trials, specifically for pediatric cardiac dysfunction. And really two of those main cell types are ones that you're going to find in our lab, which is going to be the CPCs, the cardiac progenitor cells, and also the mesenchymal stem cells. So as far as these different effects that we have from the different cell types you might use, let's take a look first at the direct differentiation. And that's going to be actually just taking cells and turning them into muscle cells, which is an extremely interesting process and a very cool technology where we can actually take human-derived cells that are human pluripotent stem cells and turn those into cardiac myocytes. Now, these cardiac myocytes are fairly immature and don't function quite the way adult cells do, but this is the one method that we have to actually make cardiac myocytes or make muscle cells to then remuscularize the heart. On the other side, we have these adult multipotent stem cells, and those are going to be our mononuclear cells, the MSCs, the CPCs, the cardiac progenitor cells, these cardiosphere-derived cells, any other stem cells that you might find in the body that actually migrate to the heart upon injury to help the heart regenerate and repair itself. And again, what we think happens with these cells is they don't actually turn into cardiac myocytes or cardiac fibroblasts or cardiac endothelial cells, cells that you might lose during disease or after an infarction. But these cells are actually releasing factors that are good for the native tissue. And some of those factors, as I said, we think are exosomes or growth factors, microRNAs, or even laying down extracellular matrix that might help the heart not form scar tissue or become fibrotic after disease. So that's actually what we've been studying in our lab pretty intently, is looking at exactly what the mechanism of these adult multipotent cells are on the heart. And we've undertaken a lot of different studies, and I have kind of our overview of the studies here. In the short term, what we do is actually grow our cells in culture. So we grow our mesenchymal stem cells or our cardiac progenitor cells, and then we collect the factors that they release. We collect those growth factors, those microvesicles, the microRNAs. Then we actually treat the native cells of the heart that we can isolate in the lab with those different factors, and we assess how it affects their function. And this has been extremely helpful for us because a lot of these cells that we can isolate in the lab, we can actually pull out of a disease state as well. So we have animals with heart failure animals that have had a heart attack. We could take those cells out that have been in that disease state and actually treat them with this media or these conditioned factors from our stem cells and see if that can improve their function in vitro. So what we do is we look at cardiac myocytes, we look at their EC coupling, which is how they actually function, their electrical excitation, contraction. We look at cardiac endothelial cells that are big in forming blood vessels. So we look at tube formation, proliferation, and migration. And then we also look at the cardiac fibroblasts, which are important for forming that scar tissue in the heart. Then on the other hand, we've looked at this from a long-term aspect too. So what we do in these studies is we actually inject our cell-based therapies into the heart and see how that affects the expression of the proteins in the heart and also the structure of the heart using echocardiography. So we just undertook a study related to this where we looked at what happens after we inject a lot of our different stem cell populations into the heart after disease. And we saw that many of these populations actually reversed the disease-altered proteome, which is actually took that diseased heart and moved it back towards a normal heart as far as the proteome is concerned, which is really interesting for us as far as trying to elucidate a mechanism of how these cells are working. So then moving on to actually some of our more mechanistic myocyte studies. As I mentioned, we isolate primary myocytes from rat hearts. So here's a nice picture of a rat ventricular myocyte. This is the T-tubules stained here. As you can see a lot of the structure of these cells. Then we load them with calcium dyes and essentially look how they handle calcium in real time under the microscope. So we use a lot of confocal microscopy and a lot of studies to elucidate the function of these myocytes at the single cell level. And then that third pillar of our research is going to be using the induced pluripotent stem cells as a drug screening platform, whether it be cardiac toxicity or novel antiarrhythmic drugs that we want to use. Again, this is a really interesting cell population that you can basically take a somatic cell and turn it into a stem cell and then differentiate that cell into cardiac myocytes. These cells are super cool. They actually start to beat spontaneously in a dish. Here's a little one that happened to look like a heart as they clump together. And what you do is essentially, these things have all the characteristics of a human cardiac myocyte, except they're fairly immature. But they're a really great system for drug screening and cardiac toxicity studies. So what we've done is develop a system where we can actually do this in a high throughput manner and measure these cells, their function with our calcium dyes. So you can see these cells loaded here and use that to assess whether or not different drugs might have an effect on the cells to correct that disease. And we've done this with a couple of different models, genetic cardiac arrhythmias, and also some environmental toxins as well, and chemotherapy drugs too. And I want to finish up by just talking a little bit about how we mentor the students here and what actually goes on during the summer program. So I'm actually going to start on the bottom here. What happens is there's a lot of hands-on learning. There's a lot of close work with mentors and the people in the lab to learn the scientific techniques. But also you get a lot of the ideas behind why you're doing the experiments and just where the field is at in general. So I know with my students, we talk a lot about moving through cardiac physiology up into where we can apply regenerative therapies as well. And understanding some of that background on how the heart works or any organ system is really critical, I think, for understanding how you can fix it then when diseases happen. So there's not just experimental things you guys are going to learn. You're going to learn a lot of therapy or theory of regenerative medicine as well. And I know a lot of the students come in and they're novices, but really the goal is for you to become the expert by the end of the program. So it's a fairly intense program. Your days are packed with learning and lab work and lectures and meetings with people. But by the end, just actually chatting with Emma before this, she took a lot of the things she learned back to her undergraduate lab to work on. So that's exactly the goal of the program, is to make you the expert in your specific field by the end of the day, by the end of the program. And I think this is a really great opportunity too, because we have a high school program that kind of runs simultaneously with your program. And in my lab, it was a really great opportunity for the undergraduate student to mentor the high school student as well. So I think there's a lot of good development of your kind of mentoring skills as well, and seeing there's a different way to think whenever you're trying to teach it to somebody after you've just learned it as well. So I think that really helps with a lot of the things we do. All right, so I'm all wrapped up there, and oops. Dr. Almeida Parada.
Awesome. Well, thank you so much, Josh, for your presentation. And as I said, we're going to have some questions and some dialogue in between. But Dr. Almeida Parada, I'm handing over the controls to you. All you have to do is just hit enter and take it away.
Okay, thank you. And thank you, Josh, for that outstanding presentation. I want to be a summer scholar in your lab.
Anytime, for sure. Anytime.
Can I come? Can I come?
Yeah,
Well done. I really enjoyed your presentation.
thank you.
Okay. So our lab focused in a different topic, or not different because Dr. Maxwell also works with children, but the goal of our lab is to cure genetic disorders. And you may ask why genetic disorders, because there's so many patients out there, 380 million patients that have genetic disorders that now they have a chronic condition, so they have to take drugs, or they actually die before even the disease is going to be diagnosed very, very early. So most of these children are, these patients are children before the age of 5. So we really want to give opportunity to these families to have a better way to treat or to cure these patients before everything starts falling apart, and then there's no therapies that can reach the goal of providing a healthy life for these children. And this is an example of two genetic disorders that impacts early on in gestation. So this is a storage disease, Gaucher disease, and you can see here ultrasound image of the liver of a patient with Gaucher disease during development. So while the fetus is in the womb of the mother, they already have these symptoms. And you can see here ascites fluid that is just accumulation of fluid that in other babies does not occur. And they have increased mass of the liver and of the spleen. And not just the liver and the spleen, is in certain cases, they also have alterations in the brain. So when we start giving drugs to these patients when they are born, there's not even the best therapies that we have right now cannot compensate for the damage that occurs during development. And here's the same with the osteogenesis imperfecta of brittle bone disease. And I don't know if you are familiar with this disease, but the children start having fractures in the bones even while they are inside the belly of the mother. So they have multiple fractures. So can you imagine a baby being born with these multiple fractures, and during their lifetime, the growth curve is, of course, not normal, and they just suffer from the pain of these bone fractures. So this is another good disease that can be targeted in utero to treat this genetic disorder prior to birth. So the focus of or the goal of our research is to look at different perspectives of cell therapies, of gene therapy, to deliver a cure for these patients while they are still in the belly of the mother. So we study AAV vectors, that is a type of virus that is right now in clinical trials for several genetic disorders. You may have heard about a clinical trial or a drug that was just approved that costs 3.5 million a shot. So you imagine if you can treat these patients before birth, then the dose is a lot smaller. Therefore, we can decrease also the price for that drug or for that treatment. And then there's the advantage of that patient never being sick, because we have cured them or treat them before birth. So we study different ways of AAV delivery and also lentiviral delivery. So these delivery agents, what they do is we use them as vectors, safe vectors, so they were virus, but we remove anything that is infectious, and we just use their vehicle to deliver genes to the body. So we include the gene that we want, the therapeutic genes. For instance, for hemophilia A would be factor 8, for hemophilia B would be factor 9, for lysosomal storage diseases would be whatever protein is missing. And so we study the safety and how can these lentiviral vectors or AAV vectors can be delivered safely, and what's the impact in development of the fetus. And it's an area that very few people, very few investigators are working on. We usually treat patients after they are born and adults, but we are not focusing treating fetus. So we have to be very careful in knowing what is safe and what's not safe for the fetus, and also what's safe for the mother. And although there's data out there for the adults, we know that it's completely different when we give a drug to an adult or to a child or to a fetus. So that is the focus of our research. We study stem cells, hematopoietic stem cells. For instance, Emma, she was working on sickle cell disease. So we are one of the ways to treat sickle cell disease is to transplant the blood cells into these patients. So Emma was studying ways of diagnosing, and a model that we have of sickle cell disease that is a sheep model, but we are also studying how we can deliver safely and with efficacy these hematopoietic stem cells to the fetus. So in an adult, bone marrow transplant is performed when we do conditioning, so that means we ablate their immune system, and then we give the blood cells into that patient after we wipe all their immune cells. We cannot do that in a fetus. So we have to study new ways of providing non-genotoxic conditioning, because what I forgot to tell you, it was that this conditioning is the same drugs that are used to treat cancer. So we cannot give those drugs to a fetus that is developing because would kill the fetus. So we are studying new ways of providing this conditioning that is not genotoxic. We also use MSCs, the same cells that Dr. Maxwell was talking about, mesenchymal cells, that we deliver genes. So we transduce these cells with these lentiviral vectors or to deliver that gene, and then now these MSCs are loaded with a transgene that can produce the protein that is missing. So we study all stem cells, all sorts of gene delivery, all with the goal of treating a fetus with a genetic disorder prior to birth. So there are several advantages of doing this prior to birth, and one of them is that the fetus, unlike a child or an adult, their immune system, the immune system, I don't know if you are familiar with, but the immune system in this case identifies what's self and not self in the case of a transplant, and rejects everything that is not self. Is what keeps us safe, right? The immune system is what prevents virus to getting us sick or bacteria from getting us sick, but also will prevent any cells that we want to deliver to the fetus that will be rejected. But the fetuses, they have a unique immunological repertoire that cells that they see, even if it's from a third party, they will become tolerant to those. So that allows us to treat the fetus in utero, and then later on using the same cells to boost their system with the same treatment, so that that treatment will go for life. We know that it's very safe because we use an ultrasound guided procedure to treat these patients. And of course, we are using a preclinical model that is an animal, but exactly the same system in which we use ultrasound and the needle, we visualize the needle, and we can deliver these therapies to the fetus. As I said, one of the diseases that we are focused is hemophilia A, and hemophilia A is a disease that in which the patients don't have factor VIII, and what that does is the patients, their coagulation cascade, what makes the blood clot, doesn't work. So what happens is that these patients, when they have the severe form of the disease, they bleed. They have huge hematomas or blood collections in the muscles. They have hemarthrosis. The blood collects in their joints. And even with the best treatments, these patients, by the age of 20, they have arthrosis like old people because the joints are destroyed by all the bleeding. So what we are devising is a cell and gene therapy that we are giving to these patients so that we can cure them prior to birth. And this is just to give you an example. Right now we have actually from the genetic diseases, hemophilia A is one of the, and B is one of the diseases that have more therapeutic options. We have concentrates, we have plasma-derived products, we have recombinant products, there's antibodies, there's many products to treat these patients. But still, we know that the chances of living a normal life will be reduced by 93% in low-income countries, and even in US, the quality of life of these patients is decreased by 33%. So that is what we want to do, is study and provide these patients with an option that makes this burden of disease disappear, and they can be healthy individuals and not suffer throughout their lives. And also we know that neonates, so the babies as soon as they are born, so these babies don't bleed prior to birth, what allows us to do the treatment, but they will bleed at birth. And we know that these patients by 1 month of age, 44% of babies that are born with hemophilia already have the bleeding. So although, like when I talked about the osteogenesis imperfecta or Gaucher disease, there's destruction during the fetal life. Hemophilia patients don't have that aspect of the disease, but as soon as they are born, they will start bleed. So if we go early in gestation, we can prevent all this bleeding while they are just infants. I was, that slide that I had, it's just all the collaborators, and that in order to do a research like that, we have to have multiple collaborations from multiple institutions, people that develop animal models, have our own models, so that we can all work together. And in the summer program, and Emma is going to speak about it, she was mentored by a graduate student, or several graduate students actually, because they have different areas of expertise. So you are right there in the middle of the research, doing the discoveries, with the faculty that will mentor you as well, that will speak with you, will show you what, or describe to you and teach you what are the problems in the field, and how you can overcome those problems with science. So Emma, it's your turn.
Yeah, thank you, Graça. So yeah, I just wanted to give you guys like a very brief overview, hopefully an insight into what it's like to be a summer scholar. My name is Emma. I'm a current junior at the University of Miami. I'm a biomedical engineering major. Previously going into the summer, I had some research experience, nothing like too crazy intensive, but I kind of identified that I was interested in regenerative medicine, interested in tissue engineering. You put that into Google and WFIRM is obviously the #1 thing that pops up. So yeah, that was primarily my goal. I thought I wanted to pursue research. I was kind of interested in a PhD, and I wanted to have a summer to really gain those research skills, figure out if a PhD was for me, figure out what those post-graduation goals are. And that was kind of a theme among a lot of the people that were in the program. Is a bunch of people were, some people were pre-med, some people were thinking MD PhD, some people were thinking straight PhD. And it is really just such a great opportunity to gain those research skills and figure out, you know, what is the role that you want to go. But yeah, just as like a really brief overview, I was super happy to work in Dr. Graça's lab. I primarily worked on designing this microfluidic device, which previous to the summer, I had absolutely zero experience in whatsoever. But the thing about WFIRM is everyone is, you know, so willing to be super interactive. You can come from absolutely no background and everyone is so willing to help. Everyone answers all of your questions, and I've literally not been in a more welcoming research environment in any place that I've ever worked. But I designed this microfluidic device that mimicked shear stress of venous capillaries, and like Grasa said, we just wanted to induce the sickling of red blood cells under hypoxic conditions. So that through the process of developing that project, I learned so much about the research process. Like I got to work with the grad students on the very basic level, figure out how much goes into before you can start anything. And then slowly throughout the summer, we, built up and we had our first prototype, and I learned, my CAD skills, needed some work, even though I was an engineer. Nothing like putting those to practice, and then fabricating the device, and then slowly, what types of experiments do we need to do before we can get to that final level, and dealing with timing. And slowly by the end of the summer, I had this moment where I'm completing the project by myself, and I kind of like looked around and I was like, oh my gosh, like I'm doing this. I came into the summer with like very limited research experience, and then by the end I'm completing a almost 10-hour experiment. There's plenty of help if I needed anything. Everyone was checking up on me and like so willing to answer questions, but like it's very validating to have that kind of experience. And then kind of like Joan said, you make a poster, and I was super, super grateful to be able to submit my poster to the Biomedical Engineering Society conference, which was in San Antonio. So at the end of the summer, we presented at WFIRM throughout the whole summer. We had also been working on an oral presentation, and we got, so much feedback on how to make a presentation, how to make a poster. And those are skills that a lot of times, like at school, you're kind of just fed to the wolves are. No one really sits down and teaches you, like, this is how you present research. And getting that like hands-on feedback was so valuable. I mean, to this day, now I go into some of my classes and I get comments all the time on my presentations of, wow, you really know how to make a slide deck. You feel comfortable talking about research. And it's from those type of experiences, like getting to talk to grad students, getting to talk to professors, getting comfortable, in that scenario. And then I was able to bring it to a conference and travel to San Antonio, present there, which then while I'm there, I get to meet all these people there. And that was all spurred by this summer. So that could not have been a more beneficial experience. In terms of like what, your day to day looks like, because I think that's a little helpful to hear about, especially as you're starting to apply. When we got there, you have 15 to 20 people from all over the country. It's, a lot of us can be a little more introverted, a little socially awkward, but like everyone's in the same space. No one knows anyone. You're all in a space that you know, like I think one person was from Wake Forest, but most people have never been here before, have never done anything. At the very minimum, you have one thing that you can talk about is like this program, your love of science, that kind of thing. And you just see them every day, and they do, and Joan especially, and Callie, they do such a good job of trying to force you together at the beginning. They organized a hike for us. We did a lunch. We got to tour the institute, and just slowly you realize you're like, okay, I'm going to be with these people for the next 10 weeks. And you make some really, really amazing friendships. I mean, at the conference, I roomed with my roommate from this summer. I still talk to my other two roommates. I still talk to a bunch of the other people in the program. And it's a really, really cool way to like, when you're not in the lab, you have these people to hang out with, to explore the city, to explore the area. Winston-Salem, I loved Winston-Salem. It was a really cool area. And then, you just feel such a welcoming environment at WFIRM. It is so open, and like a lot of universities where, your lab is like not connected to any other labs or anything else. WFIRM is a very open concept kind of building, which facilitates a lot of collaboration. It was very okay for you to just walk up to another bench and be like, what's going on there? And, your grad students that you work with know grad students in other labs, so then you can talk to them. And, by the end of the summer, I felt so integrated into the entire building. You knew people in other labs, you knew other PIs. And they do a really good job. We did seminars once a week. Dr. Maxwell and Dr. Parada's just talks they just gave are like kind of a great glimpse at what those talks kind of were like. It was like once a week you would sit down and you get to hear firsthand from these professors talk about what they are passionate about. And when else do you get to do that? Like hear firsthand from all these people. And if you're interested in research and you don't really know what you're interested in, that's a cool way to like hear a little bit about everything. And even if you know what you're interested in, it's always great to hear other things, and everyone is so welcome to like hearing you ask questions and so passionate. I mean, I pick Dr. Maxwell's brain all the time. So yeah, but it was a very open environment. And, you know, our average day, we would come in, we'd work with the grad students. We got to participate in lab meetings, which teaches you a lot about research and all of those kind of things. So hopefully that gives you guys a little overview of what we worked on. But yeah, if you have any other questions about, you know, that kind of environment, what the day in and day out was like, I'd be happy to answer those kind of questions too.
Wow. Wow. Thank you. Thank you all. At this portion, we really want to open it up. We have a nice small group here. But if you're more comfortable submitting questions in the chat, that's great. But, you know, we're here to help you. And as Emma mentioned, and it might sound corny, but it's kind of real. We really consider the people at the Institute, you know, Dr. Attal always says, once you come in, you're always family and you're always welcome back. You know, I think that's what makes the place so special. And then also, you know, just the incredible commitment from our faculty, our graduate students, and everyone. But if you have questions about the science, more questions for Emma about the experience, questions, Callie is here as well. She is the education program manager, the application, but this is the opportunity to do that now. So yeah, let's just open it up for some questions.
Hello, everyone.
Lauren, go ahead. Lauren?
Hi, sir. I didn't know if I think it's Alexandru. I don't know if he wanted to go first.
No, it's fine. It's fine. I can wait. Don't worry.
Okay, I have a question for Emma, and then I'll wait and for Alexandru to go, and then I'll ask something for Josh. Did you ever go through imposter syndrome or some type of just like a tough point within your research, and how'd you get through that?
Yeah, like definitely like the feeling of like you look around and you're like, oh my gosh, I'm only an undergrad and I've only been in this lab for a couple days, and everyone else has, you know, almost a whole lifetime behind them, specifically in this field. You definitely have moments like that where you're like, oh, how am I going to catch up? How am I going to, you feel, especially like a lot of us are very type A, you know, and are used to kind of knowing everything. But I think kind of just accepting the process that like research is not knowing everything, and, you know, being able to lean on the other people in the lab. Like something that I really appreciated is both of my grad students on the first day, they literally told me like, it sounds corny, but there are no dumb questions. Like ask every single question you have, because otherwise it's going to block you, and like we're not going to judge you. And Dr. Parada also does a really great job of that. Like I think some PIs can seem harsh even though they're not. But they really like, if everyone created such an environment of like, this is a learning experience. So once you were able to like get past that of being like, I don't care if I'm dumb, like I'm here for 10 weeks, I'm going to learn as much as I can. That was like really freeing, and that allows you to like learn a lot. So I didn't feel that vibe at all, and that's partially because of how welcoming the environment was.
Okay, thank you. That's good.
Alexandru?
All right. Thank you so much. So my question would be, how are participating students paired up with their mentor, or where can we see all faculty or all professors research projects for the summer? That would be my question.
Unmute. So, you know, Doctors Maxwell and Almeida Parada can jump in here as well. But as I mentioned at the start, we're an incredibly large institute of regenerative medicine, and we're translationally focused. So the things that Josh and Grasa were talking about is, you know, we're committed to getting these things into patients, and our portfolio is ever changing. So, we work in about 40 different tissues. And we're really involved in projects from diabetes to lung disease, chronic kidney disease, musculoskeletal, infertility, a lot neurological, a lot of cell and gene therapy approaches for, as Grasa talked about, immune-related disease, oncology. Those projects are consistently evolving. So we don't list, like some other projects, that there's a standard project in this area. We try to get you involved in things that are current, so what you can do is look at the website, and you're going to see the schedule from a couple past Summer Scholars Program, and you'll see the title of projects that students are working on. In the application, there is an area where you rank your specific interests, whether it is stem cells, whether it is enabling technologies, biomaterials. In your application in the essays, it's really very compelling. There's an area that is, what is your specific interest? You can go to the website and see what each of the researchers are focused on. They'll describe their research interests. You can see their publications, their current work. It really bodes well if you rank those things that you're interested in and then talk about. I mean, we've had students, Jake Miller is still with us. He was aware of Dr. Opara's work, who is listed on the website, the bioartificial pancreas, pancreatic islets. He was very interested in diabetes research. We saw that in his application and we made that match. But we don't list protocols of specific projects because they are ever evolving. And we want you, for the 10 weeks period, to be able to jump into a project where you're actually going to be able to carry out experiments and have some data to present during those 10 weeks. Josh and Grasa, I invite you to have any additional comments.
Yeah, I think you're absolutely right. I think it's very difficult for us, the faculty, to say, well, this is the project that you're going to work in July, because grants change, advances are made in the field. So is why I try to, and Dr. Maxwell as well spoke about areas, right? This is the area that we work on. So, if of course you are interested in biomaterials, that wouldn't be me. So you have to find someone that does biomaterials. So you have find us in the internet. There's under WFARM, all of us have a profile and what we do, and then you see the area that you would like to work, the broad area, and then you discuss. I told Emma, well, these are the different projects, and several of faculty, many faculty do that. You could work on this and that, and then you pick which one is more appealing to you. Maybe some mentors are more, they have a specific project. But I think all of us are flexible enough. I know it's 10 weeks that you can kind of say, no, I'm more interested in this area. And you can even direct that area of research to your interests and focus on that area with your mentor and your lab mentors. Josh?
Yeah, totally agree. And I think for what we usually do is the undergraduate comes in and they just incorporate into what's happened in the lab. And I don't think it gets more cutting edge than that, right? So this is exactly what we're doing right now and what's happening, and they get to join that aspect. And if there's other things they want to explore, that's certainly an option as well. But
and what we also do, once you're contacted by the end of March, and we start facilitating mentor matches based on your interests. Each mentor is a little bit different, but you start dialoguing with your mentor, and as Grasa and Josh mentioned, they'll learn more about you, your specific interests. The projects are ever evolving, and then magic happens. But if magic doesn't happen that early in the process, you are always invited to reach out to me. That's what you should do, and just say, look, I'm not really excited about this. And we have had that happen. We had that happen last summer, and we made a reassignment, and so it all worked out. But we've been doing this for a very long time, 95% of the time. And we see them in the reviews. Students will say, at first, I didn't really think this would be a good match, but we're looking at your skills too, some of the skills that you have, and trying to set you up for success. At times we just ask you once you're there to kind of hang in there for a little bit and see if that magic happens very early in the program. If it doesn't, we have a dialogue, and then we reassess and rematch. That has happened so infrequently. I've been at the Institute now almost 10 years, and it's happened 3 times.
All right, thank you so much. Appreciate it.
My pleasure. Any other questions? I want to be respectful of time, particularly our presenters. But Danielle, please.
Yes, hi, how you doing?
I'm doing great, Danielle. Where are you from?
I'm actually from Sumter, South Carolina, but I attend school at North Carolina A&T State University.
Fantastic. We actually have a lot of collaborations with NCAT and our Oregon on a Chip work. So how can we help you today?
Yes, ma'am. I actually have 2 questions at that. So in regards to the program, say if a student does get selected, in regards to housing, will there be like any provided housing, any compensation in regards to commute, or anything of that regards in general?
Great. Those really great questions, and they're important questions for undergraduate students who are coming to a new area that they know nothing about. We give, the students are housed at Wake Forest University at Ronalda campus. That is located about 4 miles. So we're in the downtown Innovation Quarter area. Many of the students bring a car, and parking, you can park at Ronalda, and they might, carpool, throw a couple bucks here and there, because you're all going to the Institute, and they might carpool together. There is also a shuttle that is provided. Now, the shuttle does not run 24/7. So you got to go early in the morning because there's only a couple runs, and there's a couple runs in the evening that take you back. But there is that option as well. There is a small housing allowance that can defray the cost of housing, and you do receive the $5,500 stipend. But Emma, do you want to say anything about the housing facilities and how that works out with transportation and such?
Yeah, I lived on the Ronalda campus. It was a absolutely amazing apartment, especially coming from a university that has kind of crappy dorms. They were really, really nice apartments. We had 4 people in each one. It was cool because everyone was living there. So that also, like most people in the program were living there. So we all hung out and stuff. Pretty much everyone carpooled because most people, like at least half of the people had a car. So most people carpooled down from the Ronalda campus to the main camp, to the 2W firm. Some people took the shuttles. The people that took the shuttles never had any issues, but we also had a group chat. So people would just kind of text and be like, can someone let me know when they're leaving? And then they would just kind of hang out at W firm until someone was leaving. And I don't think anyone ever had that many issues with it. I had a car, but I was constantly taking people, back and forth. So the apartment was great. I definitely recommend living there if you can.
You have like a 3-foot swimming pool so nobody drowned too. It's hot here.
Thank you for that. Yes.
Oh.
And also my second question, I know there's probably like a couple of hundreds of students that want to apply for these positions. And sorry if y'all already answered this question prior to, but exactly how many recipients do the W firm actually take in regards to the summer program?
Sure, good question, Danielle. The cohort is anywhere from about 18. I think the largest group we ever had was 26 students. And that is always dependent on, funding and all that. Right now, we are anticipating 20 slots to fill.
Okay.
Okay, and we receive anywhere from 200 to 350 applications, but don't ever let that dissuade you. You know, if you're interested, you know, just put together a compelling application beyond this Zoom session this evening. If you continue to have questions, anyone on this call, we did it kind of late. And so we didn't have a lot of time to recruit for interested folks. But you know, just reach out to me or to Callie, who's also on this call. Our contact is all over the application and on the website. But yeah.
Awesome. Thank you so much for the information.
My pleasure. Anyone else? Well, I have a question here that I'm going to share with the group because I actually get this kind of question from students frequently. And Stephanie asked about what my recommendations would be putting in your personal statement that would make you stand out, especially when you don't have research experience. And I'll also look toward, you know, anyone, you know, Josh and Grace's input as well. But what I always tell students is that be genuine. This is an undergraduate program. Some students may have had some prior research experience. Many students don't. The purpose for us is to expose students to research. What we really want to see, what is compelling, is a justified interest in the field. Now, sometimes, crazily enough, we receive an application, and they're all great students, you know, that they might express that they're really interested in a research experience because they don't have any, and they really think it's going to help them apply to graduate school or to medical school. Well, I get that. I mean, I get that. But why are you interested in regenerative medicine? And address your lack of research experience. Hey, you know, I'm at an institute that doesn't have a lot of research opportunities. Yeah, you know, you know, we take predominantly sophomores and juniors. We do take freshmen. We fill the whole cohort with people with all different kinds of experience, but be genuine. Also, if you have a semester where, you know, you kind of, you know, did sort of tanked on a couple things and then you came back strong, life happens. Life happens to all of us. So address it. Be transparent, express your enthusiasm. And I would say, and also I'd like to hear from Josh and Grasa, I think it's always, you know, I mentioned Jake Miller, many students, they took some time looking at faculty. They don't have to know the science. You don't have to, I mean, you don't have to regurgitate the science that they do, that they know really well, and they can punch holes all in it. I mean, that's crazy. But, you know, are you interested in, you know, diabetes, more like relate your interest to what somebody's doing? Grasa, Josh, anything else?
It's always been my requirement is enthusiasm. Grades or things like that don't matter at all. If you're enthusiastic, I can work with you. I don't want you here just because you got to tick a box for some application. Yeah, if you're excited about science and projects, and even more novice people are even more exciting because everything is so new and so fresh for them. So even better, just be excited about it.
Yeah, and even if you don't have experience, tell why you don't have experience, right? I mean, that those opportunities are not there for you, or you couldn't engage in research for such and such. And that, I think, takes you a long way, because then we know, well, this person, they don't have research at their institution, or they couldn't because they have too many doing other things that are also very interesting. So just, yeah, like John said, just be transparent. And I think being in this meeting here should gain you some brownie points, in my opinion.
I think you're right. All right, so we're going to wrap this up. We're a few minutes over, and I want everybody to enjoy their evening. And also, you know, we have holidays coming upon us, so I'm sure you're traveling to get to your families or friends. And so that's it. Thank you all so much. Thank you, Emma. Just a joy to see you again. We love our students that come, and it's also really hard on us when they go. It's like, it's very quiet. Jacqueline, you had one more question though, so go for it.
Yeah, sorry for sneaking in at the last minute, but I just wanted to make sure if we want to see like a list that probably has like most of the mentors that are going to be serving this upcoming summer, we can find that on the website, right? Because I did find a document with like 2019 mentors. I'm assuming, is that close enough to the mentors that will be there for this coming summer?
Yeah, that's close enough. And what you can do, sometimes all organization websites can be a little tricky and you have to spend some time navigating it. But each program, because, and I don't want to get too convoluted, like if you look at our T32 or summer scholars or under certain grants, you'll only have the faculty that are listed there. But we really, in this program, it's funded by so many different resources. All of our faculty have, can potentially be mentors to students, and they submit projects. Go to the website. If you look at the faculty tab, not education, the faculty listed there are potential mentors in the program. And then you can just click on their name and you'll see their research interests, you'll see their publications, you'll see a snazzy picture. Some may have been taken 50 years ago. Yeah, so you'll get lots of information. If you ever get lost and you're not finding what you're looking for, just shoot us an email. But spend a little time, grab a cup of coffee or something, and just kind of sit down near the website. The information is there.
Okay, but in like the faculty tab, that would be the place to look if
Yeah,
like,
you'll
any
hit
professors-
faculty, there will be box, and then you'll just see like alphabetical lists. It's all in alphabet, and so,
And then it would
broad
indicate if
is at
they're
the
like
top
a
of the-
mentor in the program.
Pardon?
It would indicate if they're a mentor in the program and summer program.
Doesn't indicate if they're a mentor. Our mentors, that's why it's a challenge for us to just have one list.
Oh, okay.
So all of our mentors, all of our faculty are eligible to be mentors. It depends upon the period of time, projects that they have going on that a student can be incorporated into. But it will still give you, I would still call them out in your application.
Okay.
And, we just really do our very best. And, a little braggadocio is that we're interested in supporting you. We do this program because we're committed to, exciting and engaging students in research, particularly in the area of regenerative medicine. So, we want you to have a good experience. So check it out, articulate what you're interested in. And like I said, if we do a bummer job, we can redress. But Pat, I think we really do a pretty good job, and it takes a whole team. We look at these applications and think in terms of matches, et cetera. We have your best interests at heart. We truly do.
All right, that makes sense. Thank you.
My pleasure. Anyone else?
I just want to add something to Jacqueline's. So you are going to be in a research team, right? So that is why it's not a program in which you're going to learn one technique or two techniques, or because that would be very easy to assign you. This is research. You are doing the research that the investigator is doing. So you are like a graduate student at that moment, right? You learn the ins and outs of all the problems. You are integrated in a project. And this is why it's so difficult to kind of pinpoint and say, well, you're going to be with this person and you can specifically learn A, B, or C, because depends how the research is at that point, right? Something that I can propose now, we are probably solving by then, and new questions will arise. So you are at the cutting edge of research. So it's more difficult to say specifically you can work on A, B, or C, because it's just research in flowing, right?
Well, thank you for that, Prasa, but yeah. All right. Well, this is a wrap. Thank you all again for spending some time with us. You know, just reach out if you need more information. Anyone else, any final comments before we crank it off?
John, there's a question regarding letters of recommendation.
Yeah, I thought I addressed it. You will send, there's an address in the application. It's reiterated 52 times. You will send transcripts to the Summer Scholar at Wake Health EDU, and your individuals who you identify to submit your letters of recommendation will also, they will send those to that address on their own. In your application, what you're going to do is you're going to list 2 names of individuals who you've requested letters of recommendation from, just their name and their email address. In the event, as we get close to the deadline, in the event, the onus is really on you to make sure the materials are sent. But in the event we don't get anything from them, we will drop them an email and just say you've been identified as writing a letter of recommendation for Jane Doe. You know, confirm and if this is so, and send your letter. That makes sense? Now I got another new message, 2 new messages. Oh, sounds good. Thanks. Oh, these are okay. Yeah. All right. And also because of the question will come, who to have write letters of recommendation. If you want to get off, you can jump off. If you have had a research experience, it would be great maybe if you got a letter from somebody who can talk about your you know, research chops and your ability to work in a team and those types of things. If you haven't, that's okay too. Ask one of your professors. You know, we get students from all different kinds of backgrounds, but I'd recommend certainly somebody, you know, from the STEM, from, you know, some of your engineering, science, whatever kind of courses. We also do have students who, you know, are freshmen and they're really new. They don't have a long relationship with a professor. But, you know, so many of you, gosh, I'm blown away by the talent. You may have participated in a science competition. Undergraduate students that took an externship during the summer, shadowing someone, doing something. You know, if that type of individual is better to write a letter because you're a freshman, your first, that's fine too. But probably not so great, although we have gotten them to, you know, have your soccer coach send you a letter of recommendation. And if you have more questions about that, how to be strategic, just reach out to me. All right. Well, thank you, everyone. Good to spend some time, and we look forward to receiving your applications. And as Graça mentioned, hey, if you attended this session, sure, put it in your application, and we certainly want to know that you were in here. So that's it. That's all I got. Thank you, everyone. And thanks, Drs. Maxwell, Amita Parada, and Emma, and Callie, and everyone. Have a good evening.
Thank
All
you
right,
so
thank
much.
you
Have a
all.
good evening.
Bye, Emma.
Bye.
Good to see you.